Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about ultracentrifuge. In the previous lecture we have seen what is centrifuge, what is the principle on which centrifuge work and what are the different types of centrifuge. So one of its type, that is ultracentrifuge, we will going to discuss today in a detail. In this also we will going to see the principle of ultracentrifuge, what are the advantages and disadvantages of ultracentrifuge, and what are their applications in different industries. So before understanding the detail of centrifuge, means that is ultracentrifuge, let us discuss what is ultracentrifuge, or what do you understand by ultracentrifugation. So ultracentrifugation is commonly used to purify as well as characterize low molecular weight polymers up to the multi-mega Dalton protein complexes and organelles. So it is basically used to purify, or you can say used to characterize low molecular weight polymers up to multi-mega Dalton protein complexes and organelle. Now let's see the definition. So ultracentrifuge is a sophisticated and advanced centrifuge that operate at an extremely high speed and separate smaller molecules that cannot be separated from the traditional centrifuge. So these are a sophisticated machine, or you can say device, and they have a very advanced centrifuge application, and it operate at extremely very high speed, means their operating speed is very high, and they generally separate the small molecules that cannot be separated from the traditional centrifuge. So it you can say simply it is the advanced version of centrifuge, and it works extremely at a very high speed, or you can say operate at very extremely high speed, and they separate the molecules, those molecules that cannot be separated from the traditional centrifuge. So if you look at the image of this centrifuge, so you can understand that this centrifuge look like this somewhat. If you resemble this centrifuge, it resembles to a very high technique, and if we talk about it is speed, so it operate at a very high speed, like up to 15,000 rotation per minute. It do rotation up to 15,000 1,50,000 rotation per minute. So you can understand up to which height it is operating. So ultracentrifugation, so it is a specialized technique that is used to spin sample at exceptionally high speed. This term. So the current ultracentrifuge, they spin as much as 1,50,000 rotation per minute, or you can say RPM, that is equivalent to 1 lakh grams. And if you compare it with the other units, so if you say in biocompare, it is 2019 B. So you can say this centrifugation has a very high speed, and it is a very essential device in any biology, chemistry laboratory, and it is used to for a day-to-day experiment that is going to be conducted in the laboratories, and they have a very wide application whenever you want to isolate a smaller sized particle, or if you want to study the purification of molecules or molecule complexes, so this centrifugation is used. So this is about the basic definition of ultracentrifugation. Now if you see the further, that the speed of the rotors in the ultracentrifuge can range from 60,000 rpm to that what we have discussed, that 1,50,000 rpm. Ultracentrifuge are mostly operated in more facilitated laboratories to perform more advanced operations. So these are larger in size and operate samples either in batches or as a continuous flow system. Most ultracentrifuge and are refrigerated in order to control the heat that might be generated due to the excessive heat. So this point is very important in interviews. Also, they sometimes they ask you the speed of the rotors. So if you see the speed of the rotor of the ultracentrifuge it ranges from 60,000 rpm to 1,50,000 rpm. So this is very important. Apart from this, if they ask you about its application, so it mainly used to purify different types of proteins, you can say proteins and DNA. So it has a great application whenever it purification is concerned, whenever you want to isolate any smaller molecule, right? So this is the definition of ultracentrifugation. Now comes to the very important part of this topic, that what is the principle of ultracentrifugation. So if you see the principle of ultracentrifugation, so the basis of you can say the ultracentrifugation is same as normal centrifugation. Means how? So they are used to separate the component of solution based on the size and density. Means on the basis of their size and density it generally work, and it generally separate on the basis of the size and density, right? So let us understand this part, and then we will going to discuss each principle in detail. So the ultracentrifuge work on the same principle as other centrifuge. There is nothing different. The working of an ultracentrifuge is based on the sedimentation principle that we have discussed in the previous lecture, that all the centrifugation work on the sedimentation principle, which state that the denser particles settle down faster than when compared to the less dense particle. Means so the particle that has a denser capacity or denser particles that are denser, so they settle down very fast as compared to the less denser particles under the gravity. Now comes however the sedimentation of particles under the gravity would taken a larger amount of time, and that is why an additional forces is applied to aid the sedimentation process. In an ultracentrifuge, the sample is rotated about an axis, resulting in a perpendicular force called centrifugal force that act on a different particles of the sample. The larger molecule move faster, whereas the smaller molecule move a little slower. At the same time, denser molecules are moved outward to the periphery of the tubes, whereas the less dense molecules are rotated towards the center of the tube. Once the process is completed, the larger and the more denser particles settle down, forming the pallet at the bottom of the tube. In comparison, the smaller and less dense particles remain either in the suspended, in the supernatant, or on float on the surface. So this is the principle of centrifugation. If we see the principle, so the denser particle, or you can say a structure, is the faster its sediment in the centrifugal field. The more massive biological particle is, the faster it moves to the centrifugal field. So in short, if we see the centrifugation, it works basically on the sedimentation principle, and the working of the ultracentrifugation is same, whereas that it is said that the sedimentation of particles this point that the denser particles settle down faster when compared to the less dense particle, under the gravity. So this is the principle of ultracentrifuge. And apart from this principle, once the process get completed, means when the ultracentrifugation process get completed, so the larger, or you can say the more denser particles, they settle down, and when they settle down, they form a pallet at the bottom of the tube. And in comparison to the small and less dense particle, it either remain in the suspended in the supernated, or sometimes what they do, they float on the surfaces. So on this principle generally ultracentrifuge work. So if you see this diagram or table, here you can see the type of ultracentrifugation. If you see the type, then ultracentrifugation is divided into two. The first is preparative ultracentrifuge and second is the analytical ultracentrifuge. Then preparative is further divided into differential centrifuge, and second is the density gradient centrifuge. Then further the density gradient is divided into the rate zonal centrifugation and isochronic centrifugation. So all these like whether it's a preparative ultracentrifuge or analytical centrifuge or differential centrifuge or density gradient centrifuge, as well as the rate zonal centrifugation and isochronic centrifugation, all these centrifugation we have discussed in detail in our previous lecture. So you can refer to that lecture for better understanding. Now let's move to the analytical ultracentrifuge. That we have ultracentrifugation is basically divided into two parts. So the first one is the analytical ultracentrifuge and second is the preparative ultracentrifuge. So if we talk about the analytical ultracentrifuge, that is AUC, then analytical centrifuge, as the name suggests, are the centrifuge that are used for the analysis of various particles present in the sample. So since its name suggests, it is a type of a centrifuge that is used for the analysis of various particles that are present in the sample. So particles that are present in the samples, the analytical centrifuge, they study that or they analyze that particle. So analytical centrifuge is a very versatile or robust method whenever you are whenever you are doing the quantitative analysis of the macromolecules in a solution. So we all know that macromolecules are those molecules that are very big in size. That is why they are known as the macromolecules. So this analytical ultracentrifuge, so it is a very versatile and robust method of quantitative analysis of macromolecule in solution. The third point says that these ultracentrifuge have a detection system to monitor the spinning and position of the particles in a real time to determine the sedimentation coefficient that aid to the analysis of particle based on shape, size and masses. So as we have discussed in our previous slides that this method of ultracentrifuge, they analyze any particle or separate any particle on the basis of their shape, on the basis of their size and on the basis of their masses. So the analytical ultracentrifuge is the most commonly used for the determination of properties of biomolecules like protein and nucleic acid. So it has a very wide range of application whenever you want to determine the properties of different biomolecules, for example proteins, for example nucleic acid. So whenever you want to determine the properties of proteins and nucleic acid, which type of centrifuge is used? Analytical ultracentrifuge is used. So you should always remember one thing, that analytical centrifuge, they analyze or you can say they monitor the spinning and monitor the spinning, as well as they monitor the position of the particles in the real time, right? And they determine the sedimentation coefficient. Whenever they are determining the sedimentation coefficient, they relate aids to the analysis of particles on the basis of shape, size and masses. Now moving to the next type, that is a preparative ultracentrifuge. So preparative ultracentrifuge are the centrifuge that are primarily used for the isolation and separation of particles in a sample by a process of centrifugation. In a preparative run of the ultracentrifuge, the content of the tubes are analyzed after the centrifugation period, unlike the analytical centrifuge where the analysis is done during the centrifugation process. Preparative ultracentrifuge can be operated for different types of centrifugation processes like density gradient centrifugation. Second is differential centrifugation, and the last one is the isochronic centrifugation. Particles in our samples are either separated on the basis of their density or their sizes. So preparative centrifuge are basically used for the isolation and separation of particles by the process of centrifugation. This type of ultracentrifugation also depends on the sedimentation principle, and they separate the particles on the basis of their density or sometimes on the basis of their sizes. And they are preparative can be operated for different types of centrifugation processes. Whenever you want to do means you can say in simple words, if you can say that preparative have different types other method also for the separation of particles. The first method is density gradient and centrifugation, and then it is a differential centrifugation, and the last one is a isochronic centrifugation. So these are the three types. Now moving to the next, that is difference between the preparative and difference between the preparative and analytical centrifuge. So the preparative centrifugation is a high velocity centrifuge that used in the separation of small submolecule particles, whereas whenever it is a ultra analytical centrifugation, so they uses a high velocity centrifuge, and they are used in analytical processes. Then analytical says they cannot be used in analytical processes, or sorry, the preparative says they cannot be used whenever it's analytical processes, while this type of centrifugation are used in analytical procedure. Then preparative says that they are used in pelleting small particles. Which type of small particles? Like membrane organelles and viruses, plasmid DNA and RNA. Whereas whenever it's analytical, so they are used to determine the mass, shape of macromolecules, like for example protein complexes, and rate of sedimentation of molecule. Then preparative says a large sample size can be processed, whereas analytical says only a small sample volumes less than 1 ml can be processed. Then preparative says less pure samples can be used, whereas analytical says relatively pure samples are used. So here these are certain differences that can be asked in the interview. They can be asked whenever you are giving any exams. What is the basic differences between preparative centrifuge and analytical centrifuge. So these are certain basic differences depending upon the sample. For example, preparative, they are using a very less pure samples, and whenever it's analytical, so they are using a pure samples. Okay, a large size a larger sample size can be processed, but here and in analytical only a small volume can be processed. So these are basic differences that you should always keep in mind whenever you are giving any interviews or whenever you are um, giving any exams. So these are the part of ultracentrifuge. So a centrifuge machine look like this, somewhat like a washing machine, and inside of this they have a motor. That this motor is again operating at a very high speed. They have a heavy fraction, and this part is having a light fraction. Then they have a very heavy layer, that is a side whenever it's a heavy fraction here, and they have a very light layer. They have a one bowl, and here you have to insert your feed. So these are certain parts of the ultracentrifuge you should always remember. And this type of equipment, or you can say device, is always there in the laboratory. So in your laboratory you can check this device. Now again we will going to discuss each part in detail. So the parts of centrifuge. So ultracentrifuge are provided with a variety of parts and components perform different functions. The rotors are an essential part of any centrifuge. Ultracentrifuge use all three types of rotors, namely vertical rotor, swing bucket rotor, and fixed angle rotors. So this is again very important, that ultracentrifuge have the three types of rotor: vertical rotors, swinging bucket rotors, and the last one is fixed angle rotors. If we talk about the swinging bucket rotor, so the swinging bucket rotors is most commonly used rotor in ultracentrifuge. So whenever it's a swing bucket rotor, so this type of the rotor, it is the most commonly used in ultracentrifugation. Why? Because this yield the highest concentration of particles. So what they are yielding? A highest concentration of particle. This is because the direction of the centrifugal force in a vertical rotors align with the position of the tube. So that is the reason this type of rotor is very important, and it is widely used in the ultracentrifugation process. So which type of rotor? Swing bucket rotor. Then comes the drive is the power unit which spin the rotor holding cells or tubes which contains solution of molecule or suspension particles. Beside the inchargeable electric device drive rotors, it includes analytical rotors accommodating up to 4 cells. Temperature device with a range and control from 0 to 40 degrees centigrade. So if you check the temperature, so the temperature device is ranging from 0 to 40 degrees centigrade, and you have to control this temperature device between this range of temperature, that is 0 to 40 degrees centigrade cell. And the temperature system allows the control of temperature in the system, as heat production is common during the operation of ultracentrifugation at a very high speed. So whenever the rotor is spinning, or you can say the centrifuge is operating at a very high speed, so there is of course there is a production of heat, right? So there is always a production of heat that is going to take place, and this production of heat has to be this production of the speed has to be controlled. So So this temperature system, they also allows the temperature in the system, as the heat production is common during the operation of ultracentrifugation at a high speed. Analytical ultracentrifugation also has a two-dimension, or you can say 2D compactor with a printing disk calculator interference and the absorption optical system. This optical system are necessary for the real-time analysis of molecules. So whenever you want to check, or you can say right. So analytical ultracentrifugation, it generally operate for a real-time analysis of molecule. Additionally, the gradient forming device, hand refractometers and recording spectrophotometer with a flow cell and fraction collector might also be present in the preparative ultracentrifuge. So this is an important thing to understand, that this system is operating on a real-time analysis of molecule, or giving a real-time analysis of any molecule, and additional a gradient forming device or hand refractometers and a recording spectrophotometer is also they are also having that thing. Now let's talk about the procedure or the step of ultracentrifuge. So the process of operating an ultracentrifuge might differ depending on whether it is an analytical or preparative. In general, the following steps are to be followed while performing an analytical ultracentrifugation. So the step is generally different whenever you are means for each type of centrifuge, whether it's a ultra analytical centrifuge or preparative centrifuge. So now let's discuss a very generalized step. Basically they are used while performing an analytical ultracentrifuge. So a small size a small sample size, that is 20 to 120 mm, are kept in analytical cells to be placed inside the ultracentrifuge. The ultracentrifuge is then operated so that the centrifugal force causes a migration of randomly distributed biomolecules through a solvent readily towards from the center of rotation. So the distance of molecules from the center is determined through a synchronic optical system. A graph is drawn from the solute concentration versus a squadral radial distances from the center of rotation, based on which the molecular mass is determined. So you should always keep in mind that the ultracentrifugation is operated on the basis of the sedimentation principle, and the force that is required here is the centrifugal force, and small size samples are taken that we have already discussed whenever we are discussing the differences between the analytical and preparative, and these are certain steps that are involved whenever you are doing the experiments in the centrifuge and whenever you are doing ultracentrifuge. So what are the uses of ultracentrifuge? So whenever in the first or the second slide, we have just talked about the uses of ultracentrifugation. So if you see the uses of ultracentrifugation, then preparative ultracentrifugation are used in biology for pelleting fraction of cells organelles like mitochondria, ribosomes, and even the viruses. So density gradient centrifugation uses a calcium cell gradient for separation of nucleic acid like DNA and RNA. Analytical ultracentrifuge allows the detection and characterization of molecular conformation changes due to the changes in pH, temperature, and other environmental factors. Analytical ultracentrifuge also helps in to differentiate between the assembly and disassembly of various biomolecular complexes. Ultracentrifuge are also used in the determination of densities of various macromolecules. Beside it allows the purification of various biological crude extract. So these are certain you can say uses of ultracentrifugation. So it has a great application in biology lab, in chemical lab, in a biochemical lab, and all the laboratories have this technique. Why? Because it is operating on a sedimentation principle and allowing the separation of molecules, and purification of molecule is done. And it has a great application in biology for paleting a fraction of cell organelles like mitochondria, ribosome, and sometimes even the viruses. So these are the uses. You should always know the uses of ultracentrifugation. And now let's come to the conclusion part, and let's see what are the precautions you should always take. So ultracentrifugation are subtractable devices, which is why they are used to be operated while taking a complete precautions because of its high speed. So you should always take a precaution. So while running an ultracentrifuge, the manufacturer manual should always be followed. The rotor should be handled carefully and should be routinely observed for the sign of corrosion and cracking. While adding the tubes into the rotor, the volume of the sample in the tube should be equal. This is very important point. Again, very important point. The number of tube added to the ultracentrifuge should always be balanced. If not enough sample is loaded is available, distilled water should be added to the tubes for adjusting the volume. दिस वन इज़ वेरी इंपॉर्टेंट. अगर मैं हिंदी में बोलने दे रही हूँ, अगर सैम्पल ख़त्म हो जाता है तो यू शुड ऑलवेज, इट इज़ वेरी इम्पॉर्टेंट कि आप सैम्पल की वॉल्यूम को मेंटेन करो. तो यू शुड ऑलवेज ऐड दी डिस्टिल्ड वाटर जस्ट टू मेंटेन द वॉल्यूम. So the rotor speed should not exceed the assigned value. So these are certain precautions you should always take have whenever you are operating this ultracentrifugation. So that brings to the end of the lecture. Hope you have understood the lecture. If you have any doubt, anything related to the lecture, you can simply leave the queries in the comment section below. And if I find any queries, so I'll be answering all the queries in the next lecture. And thank you for watching. Please do like, share and subscribe our channel, and don't forget to press the bell icon to get more updated. So thank you again students for watching.